Homeostasis.
Where your body likes to be.
Maintenance of a relatively
Stable
internal environment. Homeostasis is maintained by feedback loops. We have two different types of feedback loops. Each feedback loop has three main components. What is the first component of a feedback loop? Receptors. What do the receptors do?
Sense the change.
Sense a change in a variable, send that information to control center, which
Makes a plan.
processes it, comes to a conclusion about how to respond, and then sends out a command to the effectors, which
Do it.
Do the change.
bring it back to set point.
Do the
Okay.
thing.
We have negative and positive feedback. Y'all comfortable with the difference between negative and positive feedback? Okay. And are y'all comfortable with the pH scale? Okay. So the pH scale goes from what to what?
So 0 to 4.
0 to 14. We call 7 neutral because we have just as many base ions as hydrogen ions. What is the normal pH of human blood?
7.35 to 7.45.
And if my pH goes above 7.45, I am in a state of alkalosis. And if my pH drops below 7.35, I'm in a state of acidosis. Now, why is pH so important to the human body? Proteins fold up to have a very specific shape, and if we lose control of our pH, your proteins are going to do, what's the process called when they lose their shape? Denaturation. What are some reasons that they will denature? Obviously, loss of control of pH. Is that the only thing? Chemicals
And
or heat will also cause denaturation of your proteins. All right, let's bring this over. My little heads up. Okay. Okay. So, first question. We have 4 major biomolecules in the human body. What are the 4 categories of biomolecules?
Carbohydrates,
Carbohydrates, CHOs, lipids,
proteins,
no,
Nucleic acid.
Nucleic acid.
nucleic acids. There we go. We talked about having monomers. Monomers are your simplest subunits, right? What is your monomer of carbohydrates? What's that? A monosaccharide. And what is your monomers of lipids that we talked about? Well, what do you think is going to be one of the main lipids that we talk about?
adipose,
Yeah, what is the name of the molecule that makes this up? Triglyceride. And what are the monomers of a triglyceride?
Oh,
No, what are they called?
long reach.
Acid or?
A 3-carbon glycerol molecule attached to 3 long chain
fatty acids.
fatty acids.
I have
All
part of
right,
it.
so that's
I
your
had about
triglycerides.
1/4.
And your monomer of proteins is? That's your amino acids. And then your nucleic acids are going to be actually 3 molecules. We're going to have a phosphate, your phosphate, your sugar, whether it's ribose or deoxyribose, and then we're going to have our base nucleotides coming across. What chemical reaction do we use to link the monomers together to create a larger molecule?
hydrolysis, no,
Dehydration
Synthesis.
Remember, synthesis, we're making something bigger. And then if we want to break them down, we use a
Hydrolysis.
chemical process called hydrolysis, breaking it apart. All right, with that said, let's start with carbohydrates. What were the forms of carbohydrates that we studied? We just had one, monosaccharides.
Polysaccharides.
What's the one in between?
Disaccharides.
Disaccharides. And then we had
Poly.
polysaccharides. What was our classic example of a monosaccharide?
Glucose.
Glucose. And
Glycogen.
Why do we use glucose as our example? Why is it so important to the human body?
It's the one that
For
the body
neural,
uses.
What?
It's the only one the body uses. The rest get converted into it. Or
muscle contraction.
True, but why do we talk about glucose here in physiology so much?
Because the
Muscle
brain
contraction
uses it.
and
Neurons have to have glucose as their source of energy, because if you ever met somebody who is in a state of hypoglycemia, you'll notice that their neural function is not normal at that point. What's our classic example of a disaccharide?
Glycogen.
Nope.
sucrose.
Sucrose, basic table sugar, because that's going to be a fructose and a glucose molecule hooked together. And then we had examples of polysaccharides, which included glycogen. How do plants store their excess
Starch.
Starch. And what do we call it when I can't metabolize it? Fiber.
Oh.
We do not have the enzymes for that. What is the main reason humans have carbohydrates in their body? What's their primary function?
Fuel.
Fuel. Acts as fuel for the body. All right. That's carbos. Next, lipids. Let's first of all name the examples. We have triglycerides. What's another example of a lipid that's really important in the human body? Steroid hormones, cholesterol, and
Phospholipids.
phospholipids. Okay, you got that one right. Oh yeah. Okay, so triglycerides. When we eat too much of anything, it's going to get converted into a triglyceride. What do triglycerides do for us? We use them for long-term energy storage, cushioning, and
Sweat.
No, it's.
insulation. We put it in the hypodermis of our skin, and that's our primary insulation. What are steroids primarily? How do they function in our bodies? What's that?
Hormones?
They are messengers. What's examples of steroids? What kind of messengers?
Like testosterone.
Like testosterone. Think sex hormones. Testosterone, estrogen, progesterone. And then we also have our corticosteroids, which we haven't covered yet. But corticosteroids are going to be chemicals released that are going to reduce inflammation. So if you've ever heard about somebody getting a steroid shot, corticosteroid shot in their knee or in their back, they're sticking that steroid in there to then reduce the inflammatory response.
Isn't that what we were talking about with your plates?
That's one option. That's one option. And we're going to release corticosteroids from our adrenal glands. We'll learn about that when we get into the endocrine chapter. All right, what is it about a phospholipid that makes it perfectly suited for use in the plasma membrane?
Has a
Hydrophilic
Hydrophilic heads that get along great with the extracellular fluid and the cytosol, and then we have the hydrophobic tails, and that's actually what creates the barrier. So let's think a little bit about those chemical bonds. So we talked about ionic and polar, covalent, and non-polar covalent. Okay, because water is the primary solvent in the human body. If you were to analyze a water molecule, what kind of bonds are these? Nope.
Oh.
Hydrogen bonds happen in between molecules.
Oh.
What is that bond?
Polar.
Polar covalent. Because oxygen is an electron whore. It keeps the electrons around here a greater amount of time, so there's your delta negative and delta positive. Now, so this is an example of a polar covalent bond. Now let's apply this to, we have a little biracial phospholipid membrane here. This is what's going to create the barrier, right? So what kind of stuff can move into or out of a cell without permission and without a channel? Only non-polar covalent molecules. And examples of non-polar covalent molecules in your body are carbon dioxide, oxygen, and steroid hormones. Those are going to be the main ones that we will talk about. And then those that are going to have a protein channel in order to move through are going to be,
Polar.
yep, anything with the charge. And then when we talk about what will dissolve into the plasma of the bloodstream, anything with a partial positive or whole positive or negative charge. So any ionic, what's classic examples of ionic stuff?
Salt.
Salts. And then what would be another couple of examples of polar covalent things that dissolve well into water?
Glucose.
Glucose, so your carbohydrates for the most part will, like table sugar will, and yep, those are all your salts. I'm at the gym. I'm gonna have something after my workout.
Electrolytes.
Okay, those are salts.
Proteins.
Protein powder. So amino acids. Most of your amino acids are going to have enough polar covalent bond or polar covalent atoms around the outside that they will dissolve in there also. All right, so I think that's it for lipids. Yep, covered that. Phospholipid. Yep, so that's lipids. Now, what are some of the functions of proteins in our body? Yeah, everything. I just want to make sure. Are you aware of all the different functions? So they can function structurally. So your ligaments and your tendons, those are all going to be out of protein, collagen fibers. Your muscles, creating tension in your body, those are mostly all proteins. Enzymes are examples of proteins. The channels that we put into the plasma membrane are going to be proteins. Movement, antibodies, traveling immune system cells, molecules, those are going to be also. And then can we use a protein as a source of energy?
Yes.
If necessary. Not our first choice, but we could. Speaking of which, what would be our first choice if we were just to start burning energy, making ATP?
Carbohydrates.
Carbohydrates are going to be number one, and then I'm going to burn
Fats.
lipids second, and then if I have to,
Proteins.
proteins. Do we ever burn nucleic acids for energy?
No.
Not that I'm aware of. All right. Now, proteins. Your basic building block is an amino acid. So if I take, how many of them do we have, by the way?
22.
22.
Well, it's 20 that are used, but there's 22 total, isn't there?
Yeah, 20 different ones in the human body. I was like, 22? Did we get a couple extra? Okay. If I were to do dehydration synthesis and link two amino acids together, what is the name of the bond that I use to link them together?
Oh.
Peptide bond.
Peptide.
So you struggled on your lab report with that. So it is a peptide bond, and then the new molecule which is created is called a
Dipeptide.
dipeptide. And if I hook a whole bunch of them up together, then I have a
Polypeptide.
polypeptide. Okay, so proteins have 4 structural levels. What is your first structural level of building a protein?
Amino acid.
What?
Amino acid.
The specific sequence of amino acids. Since we have 20 different amino acids, Does it matter what way we put them together? Is it 1, 2, 3, 4, 5, 6, 7, 8, 9, 10? Or is it 20, 19, 18, 17, 16? Because every single amino acid is going to have a different
R.
shape
Oh.
because of the R group. All those R groups are different, so it depends what sequence we put them in. So that's our primary level. Then our secondary level is
Pleated sheets or helix.
pleated sheets or spiral helixes. So those happen within the polypeptide and give it a unique shape. What is our tertiary level of protein folding?
2D or 3D?
Okay, but what's happening? What causes that third level?
The spiral?
The folding up.
Oh.
Holding it
How
A
it folds.
single polypeptide folds upon itself. And what holds it into that shape?
pH.
Louder?
bonds?
Hydrogen bonds. That's what creates the pleated sheets. That's what creates the spiral helix. That's what causes it to fold on itself. Now, if this is my tertiary level of structure, how does this differ from my quaternary?
More than one.
More than one polypeptide associating with each other. So I'd have one here, and then I'd have another one of me right next to it, and the hydrogen bonds would keep it together. What is our classic example of a protein with a quaternary structure? More specifically, what's the molecule? Hemoglobin in that red blood cell. Hemoglobin is 4 polypeptides held together with those peptide bonds. All right, so that's proteins. And there's our primary, secondary, tertiary, quaternary. And we already talked about denaturation. All right, so what are the 3 molecules that we talked about with, what are our examples of nucleic acids? Common everyday examples. There you go, there's 2. RNA is nucleic acid, DNA is nucleic acid, and
Messenger RNA?
ATP.
ATP is a nucleic acid. All right. RNA has a pattern of base nucleotides associating with it, and DNA has another pattern. What is the pattern for the base nucleotides of DNA to match up with each other?
A and T.
A
No.
A
to
pairs
T.
with
and
T.
T.
And then C to G.
C pairs with G. And if it's RNA, what's different?
A and T
U.
stays the
A goes to U if it is RNA. So a little bit of that. Okay, so let's practice this. What is in the nucleus of a cell?
DNA.
DNA. And DNA is too big to come out of the cell. So we copy the information from DNA onto a strand of
RNA.
RNA, and then the RNA leaves through a nuclear pore and finds what organelle?
ribosome.
Ribosome. And what does the ribosome do with this recipe?
Copies the
Turns it into a
mRNA. Oh.
protein. We
Oh.
ready? This is your messenger RNA up there. We're going to turn it into the protein because that's the job of any ribosome. What do we call this process of copying DNA, the information on DNA, onto a strand of RNA?
Transcription.
Transcription, like a transcriptionist in the courtroom is going to make an exact copy of whatever is being said. And then what do we call that process of taking the recipe on the messenger RNA and turning it into a protein? Translation, okay, transcription and translation. So there's base pairing, there's transcription and translation, and that is chapter 2. Seems so much easier now, huh?